Hello, my name is Damien Fair, and I'm here to talk about the HBCD data acquisition, processing, and public access tools. I'm sorry I couldn't be there with you all today, but happy to present this talk via video. I have one disclosure. I'm a founder and board member of Turing Medical, which licenses the firm software, which is also being used for HBCD. And we started this enterprise. This is what our data flow and structure look like around data capture, processing, data management, our PII clinical warehouse, and of course, our public access portals. Over time, after we began putting the study together, this is what it turned into. Across many different universities, several different external data sources, data management areas, and of course our end goal, which is to get this data to the hands of our community. So the data management is managed by several people from both University of Minnesota, McGill, Lasso, University of California, San Diego, and Washington University. I won't be able to go through all the names of all those folks, but I'll put them up there anyway, so you just can see their faces and know who they are. The first stop in our data capture is and essentially onboarding of new participants is through Ripple Science. It also manages a lot of the external data surveys, or at least projecting them to folks in their own homes or remotely, as well as getting the proper addresses for geocoding and things of that nature. Our EEG workflows are run by Nathan Fox and Santiago Morales and their teams. They also work very closely with our team from McGill that manages Loris and has put together a nice data capture mechanism to pull all these data and organize them into the bid structure. We also capture electronic health records that is managed by teams both at the University of Minnesota and at Washington University. Those data end up in our secure data warehouse and are scrubbed and cleaned for consumption. Our data capture for MRI is done through the Fiona box. This is one of several features that we were able to replicate from the ABCD study, which helps manage and QC data coming in from our MRI scanners. Our biospecimens, our biosensors team does work in both activity monitoring as well as different sensing with regard to heart rate monitoring. Also captured by Loris. And we have several biosamples that are collected as part of the early onboarding and throughout, blood, nails, and more. All the survey data is captured and pulled together into our central database system using RedCap that is managed by the team at UCSD. And a lot of the integration of the data that eventually ends up in our environment is done through our secure data warehouse, where we organize and manage and prepare a lot of the data to upload into our data management system. Our system, it lives on an on-prem S3 system at the University of Minnesota that's maintained by the Minnesota Supercomputing Institute. Several system administrators, security individuals, and other folks are available to help organize that data. Processing of the data is federated and can be done through CBrain. You'll notice some odd pictures every now and then in this presentation because I couldn't find all their pictures on the internet, and so I generated them with AI using their names. Our central data management, project management, and system is Loris. A very large team at McGill has built a software. We have a very nice environment to help manage that software at the University of Minnesota. That is really a team effort for all data management, data workflows through this system. Our leads, some of our leads on both of this come from both Lasso, Lee McIntyre and Samir Das from McGill, who are amazing advocates and extremely good scientists in putting a lot of this work together. As you heard earlier, we have, and as you had heard earlier, we have several different MRI modalities of interest and domains of interest around structure, function, and aspects of metabolism. We're collecting T1s and T2s, diffusion MRI, quantitative MRI, again, functional MRI, and also MRS spectroscopy. To handle all this data, we've put together several different processing units or processing pipelines. Some of these you folks have probably heard of. They include knee babies or infant fMRI prep, QSI prep for diffusion. SciMRI is a commercial tool that's used for quantitative imaging that you heard of today. Functional is also processed through knee babies and also XCPD. It's not on this slide. MRS is through software called Osprey, and EEG is through software out of Maryland called MAID. All the data is organized again through LoRa's federated computing through CBrain, and derivatives are placed on our S3 buckets for consumption. Now, across these different pipelines, there are several basic principles that we've adopted for the software. One is we want maximal transparency and reproducibility. All the underlying code is on GitHub. The processing environments are available via containers. We are trying to make them as easy to use as possible. So there are BIDS apps that allow for streamlined processing. There's extensive documentation that will be available describing all the workflows and the outputs. Several reports that allow for quick quality control. We're trying to standardize with best practices using tools like nMind. We want these tools as best as we can to be community driven. So we have lots of input from several folks around our community that can also contribute to the development and maturation of these workflows. And we want them to be consistent and harmonized with our other big data sets like the ABCD and ABCC derivatives. So most of these things are actually generalizable and consistent with some of the work that's coming out of that study as well. We have been leveraging and are trying to make available as soon as we can crowdsourcing of the quality control, particularly for the derivative outputs using Swipes for Science tool that came out of University of Washington. To ensure that we have good quality data, to leverage our community to help us make sure that data is of high quality, and doing it all in a secure way. A lot of that management will be conducted through Lasso, which you'll hear about a little bit later today. Okay, so the data releases and the public releases of the data set will come in a few different forms. The first is that all of the data, all the derivative data, all the tabulated data, all the data would be organized in a BID style format on S3, accessible S3 at the University of Minnesota and my institute, the Masonic Institute for the Developing Brain. But the first stop for all users will come through Lasso. Again, you'll hear a lot more about this data platform shortly, where there's lots of features around generating a duck and proper access controls, the knowledge base, the dashboards, the data is queryable, downloadable. All the tabular data and file-based data will be downloadable directly from this platform after specific queries. Platforms being leveraged for both HBCD and ABCD, and this includes the ABCC data set that many folks are now familiar with as well. All this data is also linked up with DEAP, which was many of you are familiar with from the ABCD study, which allow for on-prem search and analytics, and several new features coming out of that exercise, which I'm sure Wes will be talking about a little bit today as well. But we've tried to make this environment even more flexible as possible. And even though this may not be ready for the initial first release, we hope it will be before too long. We're working to provide authorized and authenticated command line access to all the data, so folks don't have to go necessarily to the GUI to access the data. Data sharing tools to allow proper data sharing of individuals' own derivative data that they may want to share either with the community or with just local users. But also, we're trying to bring the federated computing and several other computing tools that will allow for analytics of the data without a need for download. That'll include access to local Jupyter notebooks, MATLAB sessions, and so on, local desktops with specific amount of resources. So a lot of flexibility for the users such that nearly any way that folks want to use the data that they'll be able to. I was tasked with trying to get through all this in 10 minutes. I hope I did a good job. I thank you all for listening. And of course, I want to just highlight all the amazing people that have contributed to everything I talked about today and what all of the data you're hearing about during this session. Thank you very much.